Okay, a little review. Yesterday, what are the four biomolecules you need in your body? We talked about one yesterday.
Carbohydrates.
Carbohydrates.
Eat your hand.
Sugars. Then we need what else?
Oh, I forget.
Sugars. Carbs.
Oh, I got one.
Kev.
Amino acids.
Amino acids, which make up proteins.
Sucrose.
That's a carbohydrate.
Saturate.
Oh wait, it's that weird one.
Lipids, fats, oils, lipids, starches or carbs. This one we don't think of as often, but nucleic acids. Okay, so we did carbohydrates yesterday. We're going to do the other three here today. We're going to start with lipids. Fats or lipids. Lipids are CHOs. They're CHO, you know, like CHO, cholesterol. CHO, just like carbo. Lipid is CHO. It usually goes 2 to 1 to 0.5. It has twice as many carbons as it does hydrogen and about half as many oxygens. Lipids are not soluble
In water.
They don't dissolve. You mix veggie oil or put butter in water, it just makes a film, it separates. It's not in the exact same order here. We talked about carbs. I'll get to that. Carbs are your energy source, right? You eat sugars, and your body goes, and you get hyper, right? Okay?
No.
What happens if you eat too many carbs? Like you store a little bit. We talked about glycogen in your liver. Johnny, listen. Your glycogen in your liver, okay? And as you need blood sugar, it breaks off little pieces.
You die from eating too much.
Mm-hmm.
Yeah, there you go.
But your body will take those extras and store it away as fats. Okay? Fats are long-term, and your blanks are different here. Long-term energy storage. Okay? Fats are long-term energy storage. It just puts it away. Puts it away for later use. Okay? What do we use it for? Chemical messengers is another, besides storing energy. Chemical messengers, like some of your hormones, are lipid-based.
No.
Some membranes, I'm talking like cell membranes.
Green cell membrane.
Cell membranes. Like next chapter when we do cells, we'll talk about the phospholipid bilayer. It's a phospholipid. We'll revisit this structure a little bit. The one we're going to draw today is a triglyceride. A triglyceride, what we would consider normal, like fat that we deposit our extra sugar as. There's actually four. I was going to say 2 parts. There's actually 4 parts because we have 3 fatty acids and 1 glycerol. So let's start with the glycerol. Very simple. You just go carbon to carbon to carbon. Handle that. 3 carbons in a row.
That's easy.
But each carbon can form how many bonds?
2, 3,
4,
5,
4.
1,
4. Look,
Eight
there's
by
1,
four.
2, 3, 4. It has
Good.
4 lonely electrons. It
Four
needs
bars.
4 pairs. So we're going to add hydrogen, 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 hydrogen. Okay, so 1, 2, 3, 4. On the inside, we're not going to put all of them yet. The first carbon we're going to put in OH.
IO.
And then we're going to connect it to a fatty acid. What are fatty acids? Fatty acids are just these big, long, what we call hydrocarbon chains, because that's basically ours. Hydro and carbons. Hydrogens and carbons. Okay, so we're going to have a carbon double bonded, okay, 2 lines, double bond. The oxygen and the carbon are sharing 2 pairs of electrons, and an OH. To a carbon, 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 dot, dot, dot. That could, dot, 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 because it could go on for a while. What do we normally have sticking off? Each carbon needs 4 bonds. So we're going to put hydrogen. Set up a little rhythm here. Hydrogen, 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 hydrogen. Okay, now, Johnny brings up a good point. How do we connect these two? How do we talk yesterday? How do we connect biomolecules?
Water.
We take out a water, that's called
Oh, the cutting fish.
Dehydration.
dehydration, to what means to put together? Synthesis. Do we see a water we can take out here? Right here. Okay. HOH, we take out a water.
What comes so?
Now this oxygen will bond directly to the carbon. Okay? So the second carbon here of the glycerol, let's show it already attached.
Wait, where does the HOH go then?
It's just water. This is all happening like in your cytoplasm, in your cells, which is full of water. So this is all happening in the water. An enzyme will come along and attach them, but take out the water. The water just like goes with the rest of the water molecules around it. Just becomes part of the solution. Okay? So here's another oxygen. Going to bond it to the fatty acid. Carbon to carbon to carbon to carbon to carbon to carbon to carbon. Dot, dot, dot. Now, sometimes organic chemists, to save them time of writing hydrogen, 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 they will just do like empty bonds like this. If you see an empty bond like this,
Hydrogens.
just assume it's hydrogens. Okay?
But we shouldn't assume. Misleading. That is misleading.
No, you can in this case.
So many
Wait,
levels.
shouldn't it be an O?
No, we're showing it put together.
Nope, I got it.
We're showing it put together. Now, the third one, I'm going to do something a little tricksy too. Watch, 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 watch. C to the oxygen. We're going to connect it to the fatty acid. And we're going to go carbon to carbon to carbon to carbon to carbon. Double bonded.
Double bond.
Oh boy.
What are those?
C's.
What is that?
Carbon to carbon to carbon. Look what a double bond. So we have a hydrogen here, 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 here just up top because those got moved up. They're sharing two. So 1, 2, 3, 4 bonds.
What's wrong with your name?
It's my smart board. This goes all.
Wait, so you should only have one double bond.
Yeah, some of them have more than one double bond. We're showing one double bond. What does the double bond do to the hydrocarbon chain?
Bends it.
Bends it. It bends it. Okay. This is where we get our.
Oh, so we should bend our thing?
Yeah, it should go. Yeah, this is not just my
I
smart
thought
board.
you were out of room.
This is not my smart board being funny on me. It actually, look, here's examples down here.
Wait, don't move it. I'm trying to do this.
Okay? This is where you ever hear of saturated fats
Yes.
and unsaturated fats?
Yes.
Saturated fats are saturated, like it rained, like Sunday night all day yesterday, so this morning the soil was saturated. It was completely full of water. If a fat is saturated, it's completely full of
Fat.
single bonds. Okay? Unsaturated, yeah, that's just the oxygen. No, we're talking carbon to carbon now. Okay? Unsaturated will have a double bond which bends them. This affects the properties of the fats.
I'm fat.
Okay?
Unsaturated fats. That's why avocado is good for you.
That's right.
Fat.
I hate
Good fat.
avocados.
If they're all single bonds, a saturated fat, it's like us all standing like this, real nice and straight, and we took out the desk, we could probably put 150 people in here. We could pack up real close and tight if we all
We
stood
could put
nice.
way more than
Like
that. You
French
could stack them
fries.
up to the ceiling.
No, I'm just talking, just standing straight. Standing.
Think French fries.
I don't think. I don't want to try. It'd be claustrophobic. And so we'd all be stacked together, real nice and tight. This makes the fat have a high melting point because they can pack nice close together. Okay? Saturated fats, your next blank there, saturated fats tend to come from animals. Animals equals saturated fats.
Wait, so
Which-
which animals equal saturated?
Yep, we get saturated fats from animals.
All blank bonds. What's that?
In a saturated fat, they're going to be all
Connected or
Single.
bonded.
single. They're all single bonds. They tend to be solid at room temperature. Think of butter. Okay, bacon grease, yeah, it's liquid, but then when you pour it off, it will solidify. Okay, will solidify. These are not healthy. These are bad. These raise the bad cholesterols, cause heart disease, atherosclerosis, hypertension.
Oh.
Unsaturated fats. Where do we tend to get unsaturated fats?
Avocados. It's an avocado.
Avocados or
Oranges.
plants. Okay, plants are where we get. Now there is one exception. We get saturated fats from palm oil and coconut oil.
Under animals. Under blankets.
Yeah.
All right.
Coconut oil. I make popcorn. I use coconut oil. It's not oil actually. It's like solid, and you scrape some out.
Milk first,
Yeah, that's the inner part of it. Okay. Unsaturated fats tend to come from plants. Plants, olive oil, okay, avocado, vegetable oil, corn oil, peanut oil. We like to fry things in. Now, those are unsaturated fats. They tend. Now, if we were all packing in here, let's say we put our arms out and our legs out, you would not get as many people in here. We couldn't pack as close together. So at room temperature, these are not solid, they're
Liquid.
liquid. We call them oils, right? Instead of like butter or fats, we call them oils.
Liquid.
Oils are much better, are a little better for you because they don't raise the bad cholesterols. There's even polyunsaturated, so there's some oils that are better than others. Trans fats you've heard about in the news lately. Trans fats are actually
why?
unsaturated fats, but they hydrogenate them, and they do things for them to make them make the oil more stable. So McDonald's can cook their fries, and again, and you can keep frying, and it's more stable. But what we've found are trans fats act like saturated fats in our body. They raise the bad cholesterol levels. So you see on a lot of menus and stuff, restaurants and stuff, advertising, and they do their fried chicken and fries there. They do not use trans fats anymore because we found that they're bad for us. We used to say, oh, they're unsaturated, it's okay. But then with research and science, we found out.
Trans fats.
What?
So everybody ate trans fats.
So everybody should eat them?
I mean, they did eat them.
They used to, yeah. You ate fries and stuff from restaurants.
Obesity.
Don't they
Heart disease.
Do they still do that?
Not many. Not many. Because they have such a bad reputation.
Who has
Is
bad
there
breath?
some double bonds?
Trans fats. Huh?
Some double bonds?
Yes. Unsaturated fats have some double bonds. They're liquid at room temperature. They're the good fats. Number 3, nucleic acids. You are missing this one.
Oh, what? Which one?
Candy broke it.
This one. I'm not that shaky, I promise.
You have that one disease where you're like,
Yeah. Nucleic acids, which are CHONP. NOPONP.
Starburst.
Carbon, hydrogen, oxygen, nitrogen, phosphorus. It stores and transmits heredity, or we will often write. There, it's getting better. Nope.
what is that, genetic?
Genetic.
Oh, yeah. I can read.
Sorry.
You should be a doctor.
Yeah, my signature. Genetic information. Now, luckily this one's already drawn for you. We did this in chapter 12. The monomer, the base unit of a nucleic acid is a nucleotide. Okay, so the base unit is a nucleotide. And I will not make you draw out all the carbons and hydrogens and oxygens and nitrogens and phosphorus because we did that already in chapter 12. The phosphate group, so I would draw it. Just draw a circle. Circle is the phosphate group connected to a pentagon, which is the 5-carbon sugar, deoxyribose.
I remember this.
Ribose. See, we did this already. Attached to remember we had pyrimidines, which were single-ringed, and purines, which were double-ringed. That's our A, T, G, and C. Those are nitrogenous bases. Okay, so if you just recognize a nucleotide as being a circle, phosphate group, a pentagon, which is the 5-carbon sugar, and a nitrogenous base, you'll be good. Okay, that we don't have to learn the C's and H's and N's and O's and T's. We did that already. These chain together. What are our main nucleic acids? Two main ones we talked about before.
Phosphate.
Based on what sugar they have. Deoxyribose or ribose. DNA
Phosphorus.
and RNA. It's our genetic material, right? And we link those together. If you remember, we built the DNA models. They're monomers. We put them together to make a polymer, this big, long strand of DNA. Put them all together.
I remember that too.
Okay, last one here. Proteins. Proteins are chons.
John's.
Oh. I think it's just that section of my smart board.
It could be anything.
Caesar.
Because it works right down here. Proteins are chon. Okay, carbon. So we have cho, chnop, and chon. This what are the main atoms in it?
Carbon, hydrogen, oxygen, nitrogen.
Yep. There. Functions, control cell reactions. By control cell reactions, I want you to write enzymes. Enzymes. We'll talk more about enzymes next week. They control how fast reactions happen. They'll bring two things and cause a reaction. Your bones and muscles are made a lot of protein. They transport substances like hemoglobin in your blood. Carrying oxygen in your blood is done by a protein. Antibodies, you talk about your immune system and antibodies. Those are proteins. Okay? What are proteins made up of? Again, we did this in the DNA chapter. AA, amino acids. Amino acids. Okay? To draw an amino acid, there's one, I'll show you. I always start with the base structure right down the middle. North Carolina Cougars.
It's actually
They lost the Super
Panther,
Bowl last year.
But it's actually Panther.
Well, Panther, Cougar, different common names for the same thing. So, North Carolina Cougars. North Carolina Cougars, off this end you're going to have an H. Over here an H. Okay, that end right here, this thing, is called the,
Yeah, we're supposed to draw that picture.
yep, is the amino group.
Ew.
Amino group. Off the middle carbon you've got an hydrogen. And down here you have an R. What element is R?
Radon. R?
Ribose.
It's not up there.
Radon.
It is not an element. R, notice they put a box around it. R stands for the remainder group. The remainder group. You have 20 amino acids that your body uses to build all the proteins it needs. How are these amino acids all different? By what is attached right here. The simplest one will just have a hydrogen. Other ones have like these big ring structures attached right here. You have 20 different amino acids. Now we talked about some of these in the DNA chapter. Glycine, lysine, methionine, phenylalanine, alanine. Yep. They are all different by what is attached right here. So R is just saying there's something attached there that makes them different. This carbon then has an OH.
IO.
And a double bonded O. This side of the molecule right here is called the carboxyl.
Yep. Like
The
that, naughty.
carboxyl, C-A-R-B-O-X-Y-L. Carboxyl group, oh, it's over here too. Carboxyl group, or sometimes we call it carboxylic acid. So you have an amino group and a carboxylic acid put together. Amino group, carboxylic acid, amino acid. That's where we get the name from, the two sides of it. Amino and acid.
What you get to put on the outside.
Like here,
Yeah, like how C then has a double bond for the O.
That's the structure of it. We just have done experiments and we know that's just what it is. Okay?
Wait, so just the R is the remaining group.
Yes.
So what would be the C and K?
That's just like the base structure of the amino acid. It's always going to be North Carolina cougar. And with all those things. Now, how do we join biomolecules together? By taking out a,
Fossil.
Water.
yep, Vasa, water.
Oh.
So we'll take out OHH. So take out an H.
Wait, so we should have 2 drawings of these?
Yeah, if you want, or just put it in your brain.
I'm putting it in my brain.
Okay, so we take out the water. Now this carbon right here will bond with the nitrogen over here.
Does that make an OH then, or not? Not that.
No, it goes straight from the carbon to the nitrogen.
Where does that O then?
Well, this gets rid of H,
No,
O,
thought
H.
bomb.
That stays there.
Does it just like move over to the left?
Yeah, this will actually kind of stick out up here. Okay. Remember, we kind of showed this. This bond right here that's forming is called a peptide
Polypeptide.
bond. Yeah, where does this happen at? The instructions are taken from the DNA in the nucleus, made into mRNA, taken out to where?
Ribosome.
To the ribosome. Remember at the ribosome we had tRNAs bringing the amino acids in. They drew them as little round spheres, and they said like MET or AA for amino acid. And then the next one came in, and then what happened? Right? It showed a little bond forming. There are enzymes that cause dehydration synthesis that cause peptide bonds. So when you get a big string of amino acids, instead of a polymer, you have a polypeptide. Okay? That polypeptide becomes the protein. Now, that polypeptide is going to be kind of curled up into a three-dimensional form. Your blank is actually structure. Structure determines function. A lot of times we'll write F cubed. F cubed, form, fits, function. Form, fits, function. So the shape of that protein is very important for what it does. There are 3 levels of shapes or structures in a protein. Right when you read the DNA, or actually the mRNA out at the ribosome, you get this big string of amino acids. That is the, and circle this down below or highlight it, that is the primary structure. See how it's just like one bead after another, one amino acid after another? Just a long string of amino acids. Next, form, fits, function. Okay, after primary structure, which is just a big long string of amino acids, we either do 2 things, or our cells do. They either will curl it. Go down here. Down. Okay, there we go. It'll either curl it, and that's what your note shows, alpha helix. Alpha helix, or it will zigzag back and forth, which is a pleated sheet. So write that where yours says alpha helix, write or pleated sheet, or sometimes this is not going to write very well.
It says A helix.
Right, alpha helix,
Omega.
or a beta sheet. It will either coil or it will zigzag.
So put A helix / beta.
Well, alpha helix is there already. See under secondary structure?
Yeah, but yeah.
So just write or beta sheet. Alpha helix is when it coils, and it will do the same thing. Like one part of it will coil, the other part will zigzag. Another part will coil, coil, zigzag, zigzag, coil, zigzag. And pretty soon it scrunches up. Then you've got the tertiary structure. Now it's like taking a paper and crumpling it all up into a 3-dimensional structure. That's kind of your final polypeptide shape.
What about coordinated?
Okay, quaternary then, we take various polypeptides and join them together. So here we have 2 polypeptides, that's quaternary. The one they're showing you in your notes is hemoglobin. Hemoglobin is 4 polypeptides. Yep, and then the little dots inside, there's the little, You don't have color notes like I do, but there's little red dots. Those are the oxygens. The heme groups which contain iron, which connect to an oxygen and carry it throughout your body. For your muscles and your brain and everything needs oxygen, right, for energy. All right, questions?